So now we have our small subunit and our large subunit. We have our messenger RNA and we have our stockpile of transfer RNAs. The first step in translation is initiation, just like in transcription. In initiation, everything comes together. So the first thing that happens is the new messenger RNA binds to the small subunit. So it lays down on that ribosome and sort of gets into position. Once there, that start codon, that AUG start codon, is going to code for that methionine amino acid. And so that first transfer RNA containing that methionine amino acid is going to come in and it's going to bind to the messenger RNA. After that, and a little bit of energy, the large subunit is going to bind as well. So now we have our translation initiation complex. We've got our messenger RNA, we've got our large ribosome subunit, and then we've got our methionine start codon. You can see that that transfer RNA is sitting at the P site. That's where we're going to grow our polypeptide chain. In elongation, the transfer RNAs that are complementary to the next 3 amino acids are going to come in sequence. So what's going to happen is that that transfer RNA is going to come in and it is going to add the amino acid to the growing chain. So that transfer RNA sits down, a little bit of energy is added, and then that peptide chain is going to be transferred from the P site over to the A site. So we're going to shift that polypeptide from the previous transfer RNA to the incoming RNA. In the next step, everything is going to shift. So this whole system sort of acts like a train on a track, and that messenger RNA is actually going to move compared to the ribosome. So everything shifts over. That empty transfer RNA is going to move over to the E site, and then the one carrying the polypeptide is going to move into the P site. That one that's in the exit site is going to be released from the system. And now our cycle can happen all over again. The next transfer RNA is going to come in, the polypeptide is going to be transferred to it, everything's going to shift over, and so on, until you reach the stop codon. So once you hit that stop codon, one of the 3, instead of a transfer RNA, you're actually going to bring in something called a release factor. So those stop codons don't code for an amino acid, but instead they encode another molecule that binds and causes the whole system to fall apart. So you can see that release factor ends up in the A position. That signals to the transfer RNA that it can release its polypeptide, and then everything falls apart. At this point, that messenger RNA can be broken down and the nucleotides reused. The ribosomal subunits are going to go bind a new messenger RNA, and then that release factor will just come back the next time that something hits a stop codon. So in a eukaryotic cell, the process of transcription happens in the nucleus. The mRNA is made, then it is processed. So translation does not actually start immediately. The mRNA is processed and then taken out of the nucleus. So a short time after it's made, transcription begins. In a bacteria, though, transcription and translation actually occur simultaneously. And that's because no processing needs to occur. So if we look at this messenger RNA here, we have a ribosome that binds and sort of chugs along the track, produces an amino acid chain, continues along, and eventually releases that polypeptide. But once this first ribosome moves over a little bit, another one can come in and move along behind it. So these are actually multiple ribosomes that are producing multiple proteins at the same time. So that's really important because you can get multiple copies out of one single messenger RNA. Now this actually can happen in eukaryotes as well. Once the messenger RNA is processed, you can have multiple transcripts going at the same time. If we look over here, we can see there's the electron micrograph picture up there, but it's a little clearer, the cartoon drawing. You have your DNA molecule, you have RNA polymerase making your messenger RNA, and as soon as that messenger RNA starts to be made, a ribosome can bind and start producing transcripts. So this is a series of many ribosomes all producing proteins as soon as that messenger RNA is coming off of the DNA. So your book gives a very nice but very busy diagram that shows a summary of this whole process. So up at the top we have our DNA molecule. RNA polymerase is making that RNA transcript. Now remember, if this was a eukaryotic cell, that would be attached to ribosomes immediately and translation would occur. But as that messenger RNA is being produced, we are going to begin to process it. So we're going to put that 5' cap on it. We're going to take out all of those introns that we don't need, add the poly A tail. Then it's going to move out of the nucleus into the cytoplasm, where the ribosomal RNA is going to bind to it. The ribosome is going to bind to it and produce that polypeptide chain. If you remember way back to chapter 4, we talked about the rough and the smooth endoplasmic reticulum. Protein synthesis actually occurs in one of two places in the cell. It either occurs actually in the cytoplasm where we have sort of free roaming ribosomes, or it also occurs on ribosomes that are actually attached to the endoplasmic reticulum. Those proteins that are in the cytoplasm are actually going to be used in that cell. The ones that are produced on the endoplasmic reticulum, on the rough ER, are going to be used elsewhere in the cell. So they're going to be produced, and then the cell is going to package them up in a nice little vesicle and distribute them or take them to the cell membrane, where they're going to be pushed out and by basically exocytosis, and they are going to be released into the body of the organism where they can be used other places. So make sure that you look at this make sure you look at this image, look it over, make sure that you understand all the parts, what all those pieces are, because although it is kind of busy, it is a great summary.